objectives of this experiment: to become familiar with the procedures of gram staining, to be able to compare the basic morphological features including the shape and the relative size, to gain some appreciation of physical diversity found in this group of microorganisms. Introduction. The application of stains that chemically react with cell material enhances the contrast between the cell and the background. Simple staining refers to the use of a single stain which is generally sufficient to display the fundamental morphological features of most microbial cells, including relative size, shape, and characteristics for groups of cells. These stains contain chromophores that chemically react with specific cell constituents or structures to visualize several unique microstructures such as flagella, endospores, and cytoplasmic inclusions. Differential stains react differently with different kinds of cells and is important in the identification of taxonomic groups. The most important and widely used differential stain for bacteria is the gram stain. Based on the gram stain, bacteria can be divided to two large groups, gram-positive and gram-negative microorganisms. The different responses to the gram stain are based on the fundamental differences in structure and composition of the cell wall. Materials used in this experiment are gram stain reagents, Hacker's crystal violet, grams iodine, 95% ethyl alcohol, safranin, staining rag, and bibulose paper.
Kak Masya dah? Nak yang bubble nak? Saya tadi. Naknya tengah pipette dah. Air bubble time-nya pipette dah tadi. So make sure kamu yang tu, tip tu, make sure dia betul-betul ketat dengan pipette. Sebab kalau tak, time kamu tarik dia tak memang 1 ml. Single kalau ni.
Kecil-kecil dia.
I feel like it's not time. Okay. Yeah. Show me this one, right? Yeah. You wait for them. So you just wait.
Oh, seminit je.
You guys can try another one, yeah?
Yeah, sure.
Banyak, dua je. Dia sampai 3 dah isi.
If-nya ada just 3.
Okey dah. Ke sana? Ada tiket. Question number 1. If the iodine step were omitted, what color would you expect a gram-negative microorganism to be? A gram-positive? The purpose of iodine in gram staining is to act as mordant to form crystal violet iodine complex. If the iodine step were omitted, both gram-positive and gram-negative microorganisms will appear pink. Question number 2. Could other dyes be substituted for crystal violet in the gram stain procedure? For safranin, crystal violet can be substituted with methylene blue. Safranin can be substituted with basic fuchsin. Question number 3. What role does the mordant play in the gram stain procedure? The mordant plays a role as a fixative to bind the dye to the cell, hence giving the color to the cell. Question number 4. What correlation is there between cell wall composition and gram reaction? Is gram reaction correlated with sensitivity to various antibacterial agents? The cell wall composition of gram-positive bacteria has a thicker peptidoglycan layer in cell wall compared to gram-negative bacteria. The ability of gram-positive bacteria to retain crystal violet is higher than gram-negative. Gram-negative is not sensitive to antibiotic actions, while gram-positive bacteria is sensitive to antibiotics.